Welcome to SWAM NPTEL course on Food Microbiology for Safe and Sustainable Food Systems. We were discussing food safety, packaging and food standards. We have already discussed food safety. Now, the microbiological considerations in food packaging. And in this we will be discussing the introduction, packaging methods, reduced oxygen packaging which will include modified atmosphere packaging or MAP, vacuum packaging, specific spoilage organisms and reduced oxygen packaging, aseptic packaging and active packaging. Introduction. In the world of food production and distribution, the significance of food packaging cannot be overstated. It serves as a vital barrier between the external environment and the food products, protecting them from various contaminants including microbial pathogens. Microbial contamination poses a considerable threat to food safety as it can lead to spoilage, reduced shelf life and even foodborne illnesses. To ensure the safety and quality of food products, it is essential to understand the microbiological considerations associated with different atmospheres in food packaging and preservation methods. Raw foods naturally contain a diverse range of microorganisms, but not all of them are capable of colonizing the food and proliferating to significant levels. Some microorganisms have the ability to dominate and grow in particular conditions, leading to the formation of a specific microbial community known as a spoilage association. The exact mechanism through which one group of bacteria outcompetes another closely related group is not always fully understood. It can depend on various factors such as the initial microbial load, the availability of nutrients, the pH level, temperature and other environmental conditions. Additionally, the interactions between different microorganisms within the food can also play a role in determining the composition of the spoilage association. Changes in the processing and packaging of fish products, even minor ones, can have a significant impact on the development and composition of spoilage associations. Different processing techniques and packaging methods can create different environmental conditions that favor the growth of specific microorganisms. As a result, the type of spoilage that occurs in fish products can vary drastically based on these factors. Furthermore, spoilage development can vary even for the same type of product, depending on factors such as geographical origin and other unknown variables. Microbial populations can be influenced by regional variations in environmental conditions, the presence of specific microorganisms in the surrounding environment and local processing and handling practices. These factors can interact with the microbial development and contribute to the differences observed in spoilage patterns. This is a pictorial diagram when to use and when to discard the food, the best before and good after. What do I do when the food has passed the date of durability, that is the expiry date? So, does it smell? You just make an examination. Does it smell, taste, look fine? If it is yes, then you can consume, and if it is no, then no. And in any case or every case when it expires, then you should not take these foods because there may be possible health risks. Packaging methods. Reduced oxygen packaging. The reduction of oxygen levels within a package effectively inhibits the growth of aerobic spoilage bacteria, as these bacteria require oxygen for respiration. This technique is commonly employed in vacuum sealed packages to preserve the quality of the contents. However, it is important to note that lowering oxygen levels may create an anaerobic environment which is conducive to the growth of anaerobic bacteria, including Clostridium botulinum. This particular bacterium is a spore forming pathogen that produces the highly potent botulinum toxin. The examples of reduced oxygen packaging techniques are vacuum packaging, modified atmosphere packaging, controlled atmosphere packaging, and sauce white packaging. Now, modified atmosphere packaging, that is with the use of gas mixtures. Modified atmosphere packaging involves altering the composition of gases surrounding the food product within a sealed package to extend its shelf life. The key gases used in MAP are carbon dioxide, nitrogen, and oxygen. The effects of different gases on microbial growth include now the carbon dioxide. The carbon dioxide acts as a natural inhibitor of spoilage and pathogenic microorganisms. By creating an unfavorable environment, it impedes the growth of these microorganisms. One of the ways carbon dioxide achieves this is by lowering the pH and making it difficult for microbes to thrive. This preservation method is commonly used in the food industry to extend the shelf life of various perishable products. Similarly, carbon dioxide helps to retain the desired texture, preventing softening and wilting of fruits and vegetables. Carbon dioxide possesses inherent antimicrobial properties which contribute to its effectiveness in food preservation. It exhibits bacteriostatic and fungistatic effects, and it thus inhibits the growth and reproduction of bacteria and fungi without completely eliminating them. This property is particularly beneficial in inhibiting the growth of spoilage microorganisms and extending the freshness of perishable foods. While carbon dioxide has a broad spectrum antibacterial effect, it is essential to note that some microorganisms exhibit resistance to its inhibitory effects. These microorganisms can still proliferate even in the presence of elevated carbon dioxide concentrations. Now, the nitrogen. Nitrogen, a widely utilized inert gas, plays a crucial role in the process of MAP. Its primary function is to displace oxygen, creating an environment devoid of oxygen that inhibits the growth of spoilage organisms that thrive in aerobic conditions. While nitrogen effectively prevents the proliferation of aerobic spoilage organisms, it does not directly impact the growth of anaerobic bacteria or spore forming organisms. Now, oxygen. In modified atmosphere packaging, the existence of oxygen can facilitate the proliferation of harmful microorganisms and lead to the deterioration of food items through oxidation. Thus, it is imperative to lower the oxygen content or establish a barrier against oxygen to ensure the preservation of food quality and prolong its shelf life. Now, effects on microorganisms, effects on aerobic microorganisms. In MAP, the reduction of oxygen levels effectively hinders the proliferation of aerobic microorganisms, including molds and certain bacteria. These microorganisms rely on oxygen for their survival, and by limiting their presence, the shelf life of oxygen sensitive products can be extended. Now, effect on anaerobic microorganisms. In MAP, elevated carbon dioxide levels play a crucial role in inhibiting the development of anaerobic microorganisms. This is a typical example of packaging of tomato slices in a MAP, which shows that it is the content in that convenient tray and a respiration controlled flexible top film. It is wrapped with that, which controls the carbon dioxide, oxygen and nitrogen content, and underneath there is highly absorbent pad which absorbs the moisture produced during the respiration. And this figure shows that if we are using MAP, then this is the extended period in the shelf life we are getting in the MAP. Now, vacuum packaging. Vacuum packaging is a preservation technique that involves the removal of air from the package before sealing, resulting in a vacuum environment. This method of preservation offers numerous advantages for packaged products, like microbial growth inhibition. Vacuum packaging creates an environment devoid of oxygen, which effectively suppresses the growth of aerobic spoilage bacteria. By eliminating the presence of oxygen, this method inhibits the proliferation of bacteria that rely on oxygen for their survival. Now, prevention of oxidative reactions. Vacuum packaging plays a vital role in preventing oxidative reactions in food products by removing oxygen. These reactions, if allowed to occur, can cause undesirable color changes, rancidity, and nutrient degradation. Because of absence of oxygen, vacuum packaging effectively safeguards the quality and freshness of the packaged food. Now, effects on microorganisms. Now, effects on aerobic microorganisms. The absence of oxygen limits the growth of aerobic microorganisms, leading to extended shelf life of the product and a reduced risk of spoilage. Vacuum packaging effectively hinders the proliferation of aerobic microorganisms, contributing to the preservation and freshness of the product. Now, effect on anaerobic microorganisms. Vacuum packaging does not directly inhibit the growth of anaerobic microorganisms such as Clostridium botulinum. Therefore, it is vital to implement additional measures including temperature control and other factors to prevent their growth and ensure product safety. This table shows the different shelf life under the vacuum packed versus non-vacuum products like beef, lamb, pork, poultry, etc. They are stored in freezer in vacuum packed, then its shelf life can be extended to 2 to 3 years, whereas without vacuum it lasts only for 6 months. For fish which is stored under freezer without vacuum pack, it has shelf life of 3 to 6 months, and with vacuum pack it has 2 years. Likewise, cheese which is stored in refrigerator without vacuum packed has shelf life 1 to 2 weeks, whereas under vacuum packaging 4 to 8 months. Vegetables like green beans, corn, cauliflower, etc., they are stored in freezers. Without vacuum packaging it has shelf life of 8 months, and with vacuum packaging it is extended up to 2 to 3 years. Likewise, apples and pears which is also stored in under the refrigerator conditions without vacuum pack, it is 2 to 4 years, whereas in vacuum packed it is 1 to 3 years, which is less than that. Bread stored in freezers without vacuum pack 2 to 3 months, and with vacuum packs it is 1 to 3 years. Cookies which is stored in freezer and pantry or the small cupboards has got shelf life of 1 month to 12 to 18 months under non-vacuum packed, whereas it is extended to 3 months to 2 years under vacuum packed. Nuts, that ready to eat cereals and spices, all these are stored in the pantry, that is the small pouches or the cupboards, which has got extended shelf life from 1 to 2 years. Now, specific spoilage organisms and reduced oxygen packaging. Specific spoilage organisms, that is SSOs, are microorganisms that have a high affinity for particular food types. They possess unique metabolic capabilities that allow them to thrive and proliferate under specific environmental conditions. These organisms have a detrimental impact on the sensory attributes, appearance, and overall quality of food products. Different food products are susceptible to specific spoilage organisms. For instance, Pseudomonas species are commonly associated with the spoilage of seafood, while yeasts like Candida and Deiberiomyces are prevalent in fruits and bakery products. Lactic acid bacteria such as Lactobacillus species are responsible for the spoilage of dairy and fermented foods. Now, aseptic packaging. Aseptic packaging involves the separate sterilization of both the food product and the packaging material, followed by filling the sterile product into a sterile container under aseptic conditions. This packaging method encompasses several key considerations. The sterility maintenance. Aseptic packaging ensures the product and packaging remain devoid of microbial contamination throughout the filling, sealing, and storage processes. By maintaining a sterile environment, it effectively inhibits the growth of spoilage microorganisms and pathogens. Barrier properties. Aseptic packaging materials offer a protective barrier against microorganisms, light, oxygen, and moisture. This barrier preserves the quality of the food product and extends its shelf life by preventing external factors from compromising its integrity. Now, effects on microorganisms. The sterilization. Aseptic packaging employs sterilization technique, that is heat treatment, to eliminate microorganisms. This process effectively eradicates spoilage organisms, pathogens, and enzymes, thereby increasing the product's shelf life. Microbial barrier. The sterile packaging material acts as a barrier against microbial recontamination during storage and distribution. This barrier is crucial for maintaining product safety and preventing spoilage. Now, active packaging. Active packaging systems are specifically designed to interact with the food product and its environment, providing additional preservation benefits. These systems incorporate antimicrobial agents or substances that can inhibit microbial growth. Antibacterial packaging materials. The integration of antibacterial agents into packaging materials creates an additional barrier against microbial contamination. These materials release antimicrobial compounds that effectively hinder the growth of bacteria, fungi, and other microorganisms. By reducing the risk of spoilage and foodborne illnesses, these antibacterial packaging materials contribute to the overall safety and quality of the food product. Now, effects on microorganisms. Now, effects antimicrobial agents. Active packaging utilizes antimicrobial agents such as organic acids, enzymes or bacteriocins which exhibit inhibitory effects on specific microorganisms. These agents assist in suppressing the growth of spoilage and pathogenic bacteria, thereby enhancing product safety and extending the shelf life of the food. Now, controlled release systems. Certain active packaging systems employ controlled release mechanisms that dispense volatile compounds such as ethanol or essential oils possessing antimicrobial properties. These compounds create an unfavorable environment for microorganisms, impeding their growth and extending the shelf life of the product. This is a pictorial representation of active packaging, where the food is product in a layer of active agents which can release some things like carbon dioxide, antioxidants, ethylene, antimicrobial agents, flavors, ethanol or preservatives. At the same time, there are certain active agents which acts as a scavenging system of the carbon dioxide, oxygen, ethylene, odor, moisture and taint. Depending upon our need, we can choose the active agent layer and pack the food in that. This table shows the active packaging systems for food, where the oxygen absorbers are used for fried snacks, nuts, high fat content powders, dried meats, cooked meat products, grated cheese, bakery products which prevents the rancidity and discoloration and mold growth, etc. Whereas the moisture scrubbers or moisture absorbers are used in the mushrooms, tomatoes, strawberries, corn, grains, seeds, fresh fish and meats, which acts as a absorbing excess moisture, decrease in moisture, condensation, absorbing excess moisture, decrease in moisture condensation, reduction of browning or discoloration. The ethylene absorbers are used in the fruits and vegetables to reduce the ripening process. Carbon dioxide emitters are used for the fresh fish and meat for extension of microbial shelf life. Antioxidant releaser are used in the fresh high fat fish and meat, powders, nuts and fried products for prevention of rancidity, whereas antimicrobial packaging systems are used for fresh meat, fish, dairy products, fruits, vegetables, grains, cereals, bakery, ready to eat meals which is used to inhibit the bacterial growth.